I have lost myself.
These are the remarkable words of the patient Auguste D., who describes her disease. What the German psychiatrist Dr. Alois Alzheimer found 100 years ago in a single patient is now one of the most frequent forms of dementia and probably one of the most terrifying diseases. Worldwide, 17 to 20 million patients are expected. Some of the remarkable findings of Alois Alzheimer were made at the Department of Psychiatry of the Ludwig Maximilians University in Munich. In the Alzheimer Museum, the university holds a beautiful collection, including photos of Alzheimer's lab members, among them famous scientists, and his collaborators and friends. The first file with the description of Auguste D's symptoms. Slides with the first brain sections. And finally, Alois Alzheimer's original microscope. Today, the University of Munich holds one of the largest centers for neurodegeneration in Germany. With Professor Haas running the division of biochemistry within the medical faculty, as well as the Munich Research Center on Molecular Mechanisms of Neurodegeneration. What are his research objectives?
If you look at an Alzheimer's disease brain section, you see immediately two different types of pathology. Huge amyloid plaques and dystrophic dying neurons filled with neurofibrillary tangles. In my lab, we focus on the major component of amyloid plaques, since we believe it initiates a deadly cascade which at the end leads to the tremendous cell loss observed in patients' brains.
Back in 1992, Dr. Haas made a remarkable finding when he was working in the laboratory of Dr. Dennis Selkow at the Harvard Medical School. Here he found that the disease-causing agent, a small and gluey protein, is produced by all of us throughout life. In other words, we are all carrying a ticking time bomb in our brains. This discovery was the beginning of modern biomedical research towards the identification of disease mechanisms and future treatment. What is Professor Haas' discovery about? The plaque component is a small peptide called amyloid beta peptide, which tends to form aggregates. This peptide is generated from a much larger protein by proteolytic processing. Scissor-like enzymes called secretases cut out amyloid beta peptide from its large precursor. Two different secretases are involved, beta and gamma secretase. Their sequential action liberates the peptide, which then can be found in cerebral spinal fluid and plasma. Upon aggregation of the amyloid beta peptide, a deadly cascade leading to neuronal cell death is started.
We believe that understanding the molecular mechanisms of Alzheimer's disease is of pivotal importance for future therapy. Based on discoveries made in my lab and many other labs throughout the world, we know that blocking secretases will slow and maybe even prevent Alzheimer's disease. In my lab, we therefore focus on the biology of secretases. We try to understand their function and biochemical properties in the disease as well as in healthy humans.
Recently, Professor Haas started to investigate Alzheimer's disease-associated genes in a simple model system, the zebrafish. Haas uses zebrafish to study the function and dysfunction of genes which are known to contain mutations causing a very aggressive form of familial Alzheimer's disease. In addition, drugs generated to block secretase function can be studied in this model for their side effects. In fact, Professor Haas and his collaborators found that inhibitors generated to block gamma secretase function caused a severe developmental abnormality in zebrafish.
Based on our knowledge of disease mechanisms, we will probably win the fight against Alzheimer's disease. I'm strongly convinced that we are on the road to successful treatment of Alzheimer's disease. However, the road to safe and efficient treatment is still very long, and we still have to discover numerous mysteries of Alzheimer's disease.